Hi everyone, good to see you again. So today's our topic will be on Entamoeba histolytica, one of the human pathogenic amoeba. Okay, let's look at these figures. Okay, this figure is showing the trophozoite of the parasites. Okay, if you can see, the trophozoite is elongated in shape, and we can see the nucleus. This is the nucleus. The round circles, dark color rings are the peripheral chromatin, and there's a central dot in the middle, known as chiosomes. So inside the trophozoite, you found that there are many dark stain color circular organelles. Actually, all these are the red blood cells that phagocyted by the amoeba during the colonization of the intestine. Okay, let's move to the next slide. This parasite is under the phylum of Sarcomastigophora, subphylum Sarcodina, that is moved using pseudopodia. Entamoeba species is grouped under the order Euamoeba. Endolimax and Iodamoeba are also grouped under this order, but they are not human pathogen. Under Euamoeba, we have pathogenic Entamoeba histolytica and non-pathogenic Entamoeba dispar, Harmani, and Entamoeba coli. Okay, under the order of Amoebida, Schizopyridia, there are opportunistic pathogens such as Acanthamoeba and Balamutia and Negrilia. Many of these are free-living pathogens. They live without the need of a host. So we are exposed to this opportunistic pathogen when we are joining activities such as swimming in a waterfall or river. This table shows the morphological description of various amoeba. You can see trophozoite and cyst stage of various amoeba. All amoeba are able to form pseudopodia, if you can see, and they have only one nucleus for their trophozoite forms. And another thing that shows difference among them are their size. So the biggest one among here, I think, is Entamoeba coli. Okay, for cyst form, if you can see, the number of nucleus different from one to another. Okay, however, one thing that consistent is the cyst form is always smaller than the trophozoite form for each of the species. Another thing that if you can notice is, if you look at the first group, there are Entamoeba histolytica, dispar, and Markovsky. All these three species show the same morphology in form of trophozoite or cyst. So it's difficult to differentiate them under the microscopes. However, still we have to differentiate them. It is important to differentiate them because only Entamoeba histolytica should be treated. Under normal clinical samples, you will be able to see the both trophozoite and the cyst form. However, for prolonged samples, maybe more than 2 hours, the trophozoite is not stable outside the host, so it will disintegrate. Whereas the cyst is more robust and could last longer in the external environment. Okay, these are the list of medically important protozoa that we will cover in this course. Our focus for this session will be Entamoeba histolytica. These are the parasites that affect the large intestine, liver, lungs, or brain. So the major clinical manifestation can be amoebic dysentery, amoebic liver abscess, or brain abscess. Okay, for this lecture, we will look into the epidemiology, morphology, life cycle, pathogenesis, clinical manifestation, and diagnosis of the parasite. Okay, Entamoeba histolytica was first discovered by Friedrich in 1873, and then the Louis Diamonds established the exilic culture, while Emily Bram she successfully distinguished the non-pathogenic form but morphologically identical Entamoeba dispar. This protozoa may be distributed mainly in the tropical areas. It is the third leading cause of death after malaria and schistosomiasis. Okay, and the transmission is occur through fecal oral route. It can be either through water or food contaminated with the cyst. Okay, oral genital transmission route has been reported among homosexual men. Okay, the disease is more severe in pregnant women, young children, and malnourished people. Okay, this table shows the three life stages of Entamoeba histolytica. They are trophozoite, pre-cyst, and the cyst stage. Okay, under a favorable environment, the cyst can undergo excitation to form trophozoites. If the environment is not friendly or stressful, the trophozoite can also undergo excitation to form cysts. Okay, so usually the trophozoite is reside in the colon, and they are irregular in shape. As they undergo encystation, the shape are changing from irregular to more and more spherical. So the size of the trophozoite and the motility is gradually reduced as the encystation goes on. Okay, trophozoite presented clear endo and ectoplasm, while the cyst is confined within a thick chitinous wall. As the trophozoite keep on growing and feeding, food vacuoles such as RBC, Weber cell, or bacteria can be seen in the clinical samples. During encystation, the metabolism rate decreases. Food vacuoles are usually not seen in the cyst form. The nucleus structure consists of central chirosome and peripheral chromatin. The same nucleus structure can be seen in both trophozoite and the cyst form. Okay, in terms of the number of nucleus, the trophozoite usually has one, but in cyst it can be 1, 2, or 4. So the storage of food in form of glycogen are only seen in the cyst, same as the chlamydia bars. Okay, now we will go into the life cycle of Entamoeba histolytica. So the life cycle consists of the cyst and the trophozoite stage. So humans get infected after we ingested the cyst contaminated water or food. The cyst will pass through the stomach and undergo excitation to form trophozoite in the intestine. So the trophozoite will move to the colon and it will undergo excitation to form cyst again. And the cyst, both cyst and trophozoite will be excreted in the feces and deposited into the environment. Okay, this figure shows the process of excitation and encystation in human bodies. Okay, first, the cyst wall is stimulated by the enzyme in the intestine, trypsin, to release a single tetranucleated cyst into intestine. Each nucleus of the metacyst is divided by binary fission to give rise to 8 nuclei. The cytoplasm of the metacyst becomes separated to produce 8 small amoebula or metacystic trophozoites. The metacystic trophozoite move from small intestine to the large intestine where it develops into normal trophozoites. So, the trophozoite can invade the mucosal tissue and finally lodge in submucosal tissue of the large intestine. The trophozoite can transform into pre-cyst stage, and the pre-cyst grows into uninucleated cyst where it multiplies by binary fissions, finally into matured quadrinucleated cyst, and it will be excreted through feces into the environment. Okay, so during the infection, if amoeba produce symptomatic disease, It can be either intestinal or extraintestinal disease stage. For intestinal disease stage, some of the trophozoite can invade mucosa and submucosal tissue and leads to intestinal disease. So if the trophozoite successfully penetrate the intestinal compartment, go into the bloodstream and reach the liver, so they can cause liver abscess in that region. Okay, now we will study the pathogenesis of the infections. Entamoeba causes intestinal and extraintestinal amoebiasis. The infection initiates when human ingested the cyst or from fecally contaminated water or food. So the cysts are carried to the small intestine where the excision occurs to form trophozoites. The trophozoite can grow and multiply in large intestine by feeding on bacterial flora. The trophozoite which remain in the large intestine develop into mature cysts and can be excreted in the feces. Some trophozoites are more invasive or virulent. They can invade the intestinal mucosa or submucosal tissue, leading to the formation of colon ulceration. So the enlargement of the ulcer produces the flask-like colonic ulcers. Some can also break through the cell wall and go into the bloodstream. Follow the bloodstream, the trophozoite can reach the liver and causes disease in localized liver. The diagnosis of amoebiasis can be classified according to asymptomatic colonization, intestinal amoebiasis, and extraintestinal amoebiasis. For asymptomatic colonization, it can be caused by either Entamoeba histolytica or DISPA. Okay, and usually asymptomatic disease is not included in the clinical management, as the infected individuals usually do not come to the hospital, but they serve as the reservoir for the pathogen, as the cysts keep on deposited into the environment. For intestinal amoebiasis, we will look into amoebic colitis, amoeboma, amoebic appendicitis, and peritonitis. Okay, this slide will show you about intestinal amoebiasis and the complication. So one of the complication is amoebic ulcers, which is the destruction and necrosis of the tissue. The ulcer can be generalized along the colon or located in a specific region such as ileocecal or sigmoid rectal region. The size can be different. It can be deep and also superficial. So another complication would be amoebic dysentery. So it is characterized by mucus and bloody diarrhea stool. Okay. So for amoebic colitis, it is the complication of amoebic dysentery. For amoeboma, it is a rare clinical manifestation, but amoeboma is characterized by necrosis, inflammation, and edema of the mucosa and submucosa of the colon, and it is usually found in the vertical segment of the colon. So if amoebic appendicitis happens, it is the rapid inflammation of the appendix. So in case of amoebic colitis, the patient will usually complain with gradual onset of abdominal pain and tenderness. The patient will tell you about diarrhea, bloody stools, and weight loss across a few days. Okay, and the patient will develop occasional fever, and frequently in colonoscopy you will find the lesion in ileocecal or sigmoid rectal region. The examination of intestine can be performed using colonoscopy. A camera will be inserted to the body through anus. The camera is usually equipped with the ability to collect biopsy as well. This figure shows the ulcer in the colon area. Okay, the biopsy sample can be sent for histopathological examination. Under the microscope, typical flask-shaped ulcer of the intestinal amoebiasis can be seen. Okay, so this is the volcano mouth regions. So with white base and irregular in the surrounding region and slightly elevated edge. So in case of extra-intestinal amoebiasis happen, it usually happen in the liver, lungs, or brain, according to the most reported cases. So amoebic liver abscess is the most reported cases. So for extra-intestinal amoebiasis, amoebic liver abscess is the most commonly reported cases, and this is predominantly happen in the male. Okay, and other complication of extra-intestinal amoebiasis is depends on where the trophozoite travel through the bloodstream. So other than the liver, follow the bloodstream, the trophozoite can form complication in the skin, in the lung, in the brain, or stomach. So as you know, amoebic liver abscess is the extended version of intestinal amoebiasis. The trophozoite successfully penetrate the intestinal cell wall and get into the bloodstream. So the amoeba will get to the nearest organ, which is liver, through hepatic portal vein. So the first lesion will be in the liver organ. So the patient will usually presented large and tense liver. So if abscess is formed, it's usually small, but slowly it will increase in size. Okay, so to confirm the diagnosis, usually we will perform fine needle aspiration. So if we withdraw the liquid from the center of the abscess, we will be able to see anchovy sauce appearance liquid. And usually the abscess is happen at the right upper quadrant region. Okay, this CT scan is showing two lesions in the liver. So there's an attenuation lesion at the right lobes with surrounding inflammation, which is consistent with the characteristic of amoebic liver abscess. Okay, now we have come to the diagnosis of intestinal amoebiasis. So for stool examination, basically we would like to demonstrate the presence of the parasite in the stool samples, whether it's the trophozoite, cyst, protein, or the parasite-specific DNA. So for microscopic examination, it remains the gold standard. So what we can do is we can use saline wet mouse or iodine wet mouse, or we can also perform trichome staining. Trichome staining is the more advanced microscopic method as it stain using 3 different colors. It will demonstrate the morphology better. So for stool culture, it can be performed, but it is not so accurate and it is time consuming. It will usually require 8 to 10 days, which is not practical nowadays. So if the cyst count is low, we can also use concentration technique like formalin ether or zinc sulfate concentration techniques. If the clinical sample show positive for the cysts or trophozoites, we can do confirmation tests like stool antigen test, which specifically reveal the presence of Entamoeba histolytica lactin antigen. We can also use blood tests to diagnose intestinal amoebiasis, but the common finding will be moderate leukocytosis. However, this finding cannot be generalized and is not specific. Okay, serological method that can be used to detect the presence of intestinal amoebiasis include indirect hemagglutination assay to detect the presence of antibody, indirect fluorescence antibody test where we will code Entamoeba on the slide, and then we will detect if the antibody is present in the serum sample, it will show fluorescent. Otherwise, it will be the color will be dim. Okay, another common method that we can use is ELISA. Okay, for the diagnosis of extraintestinal amoebiasis, the most commonly reported cases would be amoebic liver abscess. For an abscess, we can use fine needle aspiration to aspirate the pus in the center of the abscess. And then the liquefied pus enables us to check for the trophozoite under microscopes. So for liver biopsies and histology, we can obtain sample using fine needle aspiration as well. And the small tissue that we have extracted, we can perform histological staining. We can perform either acid shift staining or H&E staining to demonstrate the presence of trophozoites. So in case we can't perform invasive method due to the limitation of equipment or whatever, the indirect method that enables us to diagnose extraintestinal amoebiasis is the detection of the presence of high antibody titers. Okay, usually the accuracy of the test is 99% sensitivity and 90% specificity. So if we are able to obtain the pus aspiration, we can also detect the presence of DNA, parasite-specific DNA in the pus samples by using PCR. Okay, this picture shows the various forms of trophozoite in the clinical specimens. Okay, these two are the trophozoite that we see under microscopy. Okay, this is the elongation form of the trophozoites. We can clearly see the pseudopodia, and there are many food granules inside the trophozoite. It could be RBC, it could be bacteria, it could be anything. Okay, but over here we can't appreciate the nucleus. Okay, over here we can see that the trophozoite is in round shape. It is in round shape. It might be due to prolonged period outside the host. So slowly the metabolism rate reduces and the trophozoite become fully round and disintegrated. So over here we are able to appreciate the nucleus structure of these trophozoites. Okay, this is a stained trophozoites. We can see the irregular shape of the trophozoites and a clear nucleus and the central chiosome surrounded by the peripheral chromatins. And I think this stain is trichrome stain, 3-color stain. Okay, this is also a trichrome stain specimen. We can see they are represented in the trophozoites and the nucleus is appreciated. Okay, next. This is another version. This is a little bit round, and this is the nucleus with the central chiosomes. And all these black stained organelles, all these might be RBC. Okay, these are tissue stain samples. We can see there are many. These are the trophozoites, and the blue stain one could be the nucleus. And there are many pink stain organelles that most likely is the RBC that phagocyted by the trophozoites. Okay, this slide show various cyst form of the parasites. Okay, they can be stained by iodine, trichome, or saline wet mount, or ion hematoxylin stain. Okay, this is the iodine stain samples. So this cyst is slightly oval. There's only one nucleus, and there's a central chiosome with peripheral chromatins. Okay, next. Okay, these are trichome stain samples. So there's only one nucleus being appreciated. Okay, it contains a large. If the black stain one are the glycogen vacuole. Okay, there's another young cyst in the field contain round chromatoids. Okay, that partially obscure the glycogen vacuole. Okay, this is a matured and immature cyst. This is the matured and immature cyst. Okay, this one contains two nucleus. This one has only one nucleus. And these are the chromatoid body. Okay. Okay, this is iodine stain samples. Okay, these are beautiful samples because it is quite intact. Okay, the cyst is usually round, but if we use any staining solution, it might distort the morphology of the cyst. Okay, so we can see here, these are the brownish thing are the glycogen vacuoles, and this is the nucleus with central chyosomes. Okay, this is a quadrille nuclear form of the cyst. We can observe one nucleus, two nucleus, three nucleus, and four nucleus. The chromatoid bar is very thin, but we are not sure whether this is Entamoeba histolytica, Dispa, or Markovsky's. Okay, this is ion hematoxylin stain cyst. There are three nucleases in this cyst. Okay, the fourth one is the centrally located and covered by the chromatoid body. This is the chromatoid body. Okay, that's all for today class. Thank you for your attention.